My name is Kakani Katija, and I'm really excited to be able to present and share with you some of the work I've been working on with a number of institutions and collaborators over the past couple of years. I really want to thank the session organizers, the SICB organizers, the executive board for SICB. It's been really, really challenging, and it's amazing that we're able to pull off, a meeting like this in such challenging times. So thank you for that. And I also want to thank you for coming and observing and learning a little bit more about some of the stuff we've been working on. And like I said, this is a multi-individual, multi-institutional effort that includes researchers at Mbari, Sea Vision AI, and the Ocean Discovery League, where we're looking to open source tools and data sets to enable artificial intelligence to help us explore and study, discover life in the ocean. And this question is actually really important to me as I was trying to put together this talk. Because you can imagine with the SICB audience, it's really important for me to make this connection, right, between what I'm trying to do with ocean exploration and discovery and how that can be beneficial or valuable to the SICB community. And so the question is, how or why is ocean exploration and discovery vital to integrative and comparative biology? And the short answer is that I think, as well as a number of other authors, think that there's a need to look beyond model organisms in laboratory environments, because there's a number of different questions that we might have about these kinds of observations. So how representative are these organismal models to the rest of life? How do laboratory lives of model organisms, animals that we're able to study really well, like how do those lives in the lab really compare with their relatives in the wild? And what I also want to bring up too is the fact that, non-traditional systems produce puzzles and paradoxes, which then help us define functional limits for life with diverse systems. And this is true for both terrestrial and aquatic environments. But what I want to posit, or at least argue, right, is that the ocean is a pretty remarkable place. The fact that animals there, animal diversity is the richest in the ocean. Marine systems contain representatives of 28 out of 35 animal phyla that exist today. And there's a real potential for us to discover truly new solutions in biology and for society if we look to the ocean and its inhabitants. But there's a massive challenge, right, in us to be able to do this, right, to study and observe life in a meaningful way, right, to our community within the ocean. And I just want to give an example of this in terms of just really basic observations of life, a point observation of different locations within the world's ocean. And this is a paper that was published in October 2021, where the authors, many of them together, collated or aggregated all of the observations that have been done to globally observe marine life for a long duration of time. But what I want to point out is, like, from one of the figures in their paper, what this is the spatial coverage of known active as well as long-term biological observations globally, where the colored areas indicate biological observations that are either identified from surveys only or from data sets that you can find in these global data repositories like the Ocean Biodiversity Information System, or OBIS, and the Global Biodiversity Information System, GBIF. And what you'll note, the main takeaway from the paper, right, is that about 7% of the ocean's surface area is covered by long-term biological observations. 7% of the ocean surface, never mind that the ocean represents 70% of the planet's surface. And now add to that the fact that, the ocean isn't just a surface, right? It's represented by massive volume. And what I like about this animation is it conveys kind of the scale of this problem, where on the left in yellow, we have the Statue of Liberty, and on the right is the Rachel Carson, which is one of Mbari's research vessels. And when we try to characterize the size scales, right, for the upper ocean, which are the lighted upper surface waters, the depth of that region is approximately 200 meters. So that's roughly two Statues of Liberty stacked on top of each other. But when we think about the ocean, though, it's a massive volume. So in addition to the sea surface or the upper ocean and the sea floor, there's this vast region called the ocean's midwaters that connects the two. And it's this region where, the midwaters comprise 93% of habitable volume on the planet. And the ocean in its entirety, we've only about explored less than 20% of that. Now, these numbers come from NOAA. I've had conversations with other researchers on this particular issue, and the number that these other groups come up with is pretty significantly smaller than this. But then to add on top of that, the fact is there's estimates that anywhere from 30 to 60% of life in the ocean is undescribed. So, right, not known to science. And that it also takes about 21 years between the species discovery, the first time we've actually observed the animal, until it's described. And I'm only talking about descriptions, right? I'm not even talking about behavior or ecology. This is a massive bottleneck that if we can address, we could potentially be able to expand the sources or the options for model organisms in the ocean and do this within our lifetimes. And there's a number of different ways that we can observe biology in the ocean. And one of the modalities that's really starting to come to the forefront is imaging, because imaging allows us an opportunity to directly identify animals, directly quantify animals, and do that in a really non-invasive way. And I like to describe our data challenges through the lens of what we've been experiencing at MBARI, or the Monterey Bay Aquarium Research Institute. And that, imaging is very, very rich in information, but it's extremely expensive in terms of data storage as well as data processing, because it requires people to look at that video. And so while we started in the late 90s with a single remotely operated vehicle that had a video stream or video feed, we're now fast forwarding to now where we have multiple of these vehicles or these assets that collect video data, not just one video data feed, but a number of them. The other thing too is we're starting to turn towards autonomy, right? If we want to be able to discover and observe life throughout the volume of the ocean, we really need to think about automating. And so we're using autonomous vehicles that have imaging systems or imaging sensors on board, including ones that just walk along or roll along the bottom, like the benthic rover you see on the bottom. And that particular robot has been deployed in the Monterey Bay area for more than 7 years at this point. But then there's also this need, right, not only to observe things like in a binary fashion if it's present or absent, but then researchers are also trying to do things like measure size spectra or biomass. And so in order to do those kinds of measurements, you really need stereo data or multiple cameras to collect that information. And so there's an example of this where on the left, we have a benthic lander that has multiple camera systems. And then on the right, we have a time-lapse camera that has multiple camera systems. And then on the top is an underwater vehicle called Mesobot. I gave a presentation at SICB a couple of years ago describing it, where this vehicle uses stereo cameras to automatically control the vehicle to track objects like animals in the water column for over a period of time. So all of these different, challenges whereby we're collecting lots of video data, we're deploying it and using it in a number of different ways. And, how then do we scale up our observations without trying to automate the process of analyzing this information? And so I just wanted to share with you some examples of what this underwater visual data might look like. So this is video collected by one of our remotely operated vehicles near the seafloor, near the wall actually of the Monterey Canyon. And in order to understand and characterize the underwater environment, researchers collect image and video data to catalog the geologic features and life that lives there. And so as you can tell, right, the visual data like this video are incredibly rich resources, which yield information about substrate type, animal species, abundance, and relative distributions, which are important quantities for measurement. And this is an example of a video that's collected in midwater and in the water column. And so this is footage collected from I2Map, which is an autonomous underwater vehicle. And what happens during a midwater transect is you move at a constant speed for a constant duration, recording video the entire time, and then you repeat those transects with depth so that you can, catalog variability of the organismal community in these midwater locations. And if you're interested in trying to understand animal distributions with time, be it daily or hourly, trying to not only collect this data, but then process it in a repeatable and effective fashion is really, really challenging. We're very fortunate in Mbari where we have a room full of video lab technicians, the taxonomic visual data experts that can process this information. But this is not a scalable resource. And if we want to achieve full exploration of the ocean so we can study non-model systems, we need to crack that net. And so where we're at, right, is this challenge. We very quickly realize that as more vehicles and cameras are used to discover the depths of the ocean, we find ourselves just drowning in data with limited resources and taxonomic expertise to fully process what's in it. And so how can we tackle this data deluge and automate this process as much as possible? And so this is why, for the last couple of years, I've been trying to answer this question with our collaborators is like, can artificial intelligence address our data challenges? And when I talk about artificial intelligence, that term is super broad. And artificial intelligence describes all programs with the ability to learn and reason like humans. And machine learning is actually a subset of AI or artificial intelligence and includes algorithms with the ability to learn without being explicitly programmed. And then a subset within machine learning is deep learning. And this kind of technique uses multi-layered neural networks that learn from vast amounts of data. So you've heard the term big data before. A lot of the community are talking about deep learning, the need for lots and lots of data or information in order to train algorithms. And what's really interesting is that over time, deep learning looks to be the approach that has the greatest potential for a number of different applications. Not only in the case of trying to identify the difference between humans and cars and stop signs and signal lights, for applications related to autonomous self-driving vehicles, but also have the potential for helping us determine differences or classify the differences between different kinds of animals in the ocean, and in some cases to the species level. And the reason why, I initially started to get involved in this problem, because I definitely don't want to say that I want to solve everybody's visual data processing problems. But my entry point into this was the fact that when we were doing work with the Mesobot, which is, you can see here in this video, the Mesobot was designed to autonomously track animals in the ocean midwaters. And this is a demonstration of the vehicle tracking an object that we're moving around in Mbari's test tank facility. And while, our algorithms that we've developed and we trialed in the test tank worked great, when we went to sea, right, the situation completely changed. And this is because of, interestingly, biology. And so what I'm about to show you is a video that I think I shared a couple years ago, but I think it brings the point home. Red circles indicate potential targets that we can track. And the blue filled dot indicates the target that we are currently tracking. And so in this video, what you're seeing is a blue dot where we're tracking a salp. And then very shortly after this, you'll notice that a squid comes by, literally bumps into the animal. And then the tracking gets transferred onto the squid and it changes the vehicle behavior. But then shortly after this instance, we noticed then that the squid wound up being joined by all of its friends. And then somehow, some way, we were able to resume tracking this salp. But imagine that the science goal was to track a single individual for 24 hours, because it really is, that's the science goal. And the disruption of tracking on another animal would mean that the mission failed. And so while we might be lucky here and are able to continue tracking the salp after the squid interference, that may not always be the case. And so if we had an algorithm or algorithms that could detect or distinguish between organismal targets, we can at least continue tracking when more than one animal appears and ignore when animals or multiple animals emerge in our field of view. Another use case that's also near and dear to my heart, and Bruce Robeson, who's a senior scientist in Mbari, is trying to understand the ecological impact and the function of mucus houses created by giant larvaceans. And so one of the questions is, how do we distinguish between animals and mucus house? And in this case, you saw the animal swam away from the mucus structure. And there's scientific value in tracking either target. If we tracked the mucus house or the snot palace, we could understand something more about the carbon cycling process. If we track the animal, we might be able to say something about its behavior. How often are they creating or building their homes? And so again, being able to distinguish between these different features as well as they change over time is going to be important And machine learning may provide that mechanism to distinguish it. And, I'm grateful to the National Science Foundation for supporting some of this early work that has also then led into the next phases of the project that I'm sharing today. And so artificial intelligence, or deep learning specifically, requires a lot of data. And data can be divided into two types. Labeled, where we know everything that's in it, like a human has actually looked through that information or the data, and then unlabeled data. So this is data where we don't know what's in it, data that we want to make predictions on in an automated fashion. And labeled data is then used to train algorithms and test them, evaluate how well they perform, and then use to, then lead to the next stage, which is to automate data processing. But what do I mean by unlabeled versus labeled data? And so this is an example of an unlabeled image where we know, looking at it, at least I do, I'm sure a lot of people here, this is a jellyfish, Aegina. It's what we call iconic view, so it's very zoomed in, it's centered. It's clear that there's only one thing or one concept in this image. And that makes it, that's great. But in instances like this one, and this happens more often, the fact that is, if you are someone or a machine that's never looked at these kinds of images before, you might, it's hard to know which is the jellyfish and which is the other animals or the other concepts. And so to label an image, that means you not only have to name or classify the object that's in the image, but you also have to create a bounding box or localization. And this means doing this not only for one object, but all of the objects in these images. And so you can imagine it takes a lot of time to create labeled data that can then be used to train algorithms. And so once you have your labeled data, though, you can take a subset of it, trains an algorithm, and then the remaining amount of the labeled data, you can test your algorithms, evaluate how well it works. And if you're not happy with the performance of the model, you can generate more labeled data and then repeat this process. And at some point, once you've done your evaluation, if your model works great, then you can apply it to your unlabeled data and then run inference or get predictions, where then you can use that information for your research. But the massive bottleneck for everybody that does deep learning is labeled data. How do you generate labeled data that can be valuable? And in our case, how can you have enough labeled data that can be used to describe all the animals in the ocean down to the species level? And the ocean, right, is a massive volume. So we're talking like a global representation of ocean life using imagery, but relying on labeled data. And so this is why we built FathomNet. FathomNet is a public, open source underwater image database for labeled data so that the community can contribute their data and use it to train machine learning algorithms. And so FathomNet provides imagery and metadata for ocean life and includes additional concepts like equipment, debris, and other geological features. FathomNet was founded by MBARI, Sea Vision AI, and the Ocean Discovery League. And FathomNet currently contains visual data from MBARI, NOAA's the Office of Exploration and Research, as well as the National Geographic Society using their drop camera system. And what we're looking forward to a number of other contributions coming from the community, but we've only just launched the website or the database in beta last fall. So we're learning as we go. The beta version, like I said, was launched. This is the landing page of FathomNet, and you can find it at www.fathomnet.org. And I think the main take home is that FathomNet is not just a database, but as an open source ecosystem. So the database can be accessed via the website at fathomnet.org or via an application programming interface or API. At the website, you can visually explore the data. You can view metadata that's associated with all of the data submissions. And I should also add that FathomNet has a lightweight annotation tool that can be used to augment existing labeled data in FathomNet. Another thing is we spent a lot of time developing tutorials and how-to articles found on Medium and YouTube channels with also our code and issues that are tracked on GitHub. So again, everything is open source. And then for those using FathomNet data, we also ask them to add their trained models to the FathomNet Model Zoo on our GitHub so others can use these models to process their visual data. And finally, what we're trying to do, though, is leverage widely available open source tools for AI model architecture and training. And I have to say, this community, the computer science or computer vision community, is phenomenal when it comes to open source tool generation, sharing, and disseminating those tools elsewhere. So we're trying to fill the gap where we're missing labeled data and we're missing these models that can then be used by the community for various projects. And so I wanted to really quickly finish up and answer this question about, can these open source data and tools address our challenges? And the answer is a bit mixed at this point. It's still a work in progress, but the future is very, very bright. I want to start by showing you some preliminary data using FathomNet, training a machine learning algorithm and deploying it on that benthic video data set I showed earlier. And you'll notice the different colors correspond to different individuals, but you can see we're getting positive hits for Paragorgia, Swiftia, Solus, Asteroideae, a number of different kinds of animals. And so what we're trying to do now is figure out ways for a human expert to engage with this data and quickly verify and correct it to create a data processing pipeline. And then here's an early example of an output using a machine learning model, although this time trained on FathomNet, but using classes of animals that are commonly found in the water column. And you can see that we're getting detections. Some of the classifications aren't completely accurate, but again, it's a work in progress. We need more labeled data. We also need more human verification to ensure that the outputs are accurate. But now remember that question that I had earlier with Bruce, our interest in trying to track these kinds of animals that are changing their shape or changing their state. And so we can demonstrate how we can distinguish between a larvation, which is indicated in this green box, and the mucus house, which is indicated in yellow. And so what you'll notice is as the animal leaves the mucus house, we get a really strong signal indicated by that green bounding box that separates or distinguishes the animal from the house. And so what's really exciting is that these kinds of algorithms can be platform agnostic, so that as imaging systems are incorporated into other vehicles or new vehicles are used, we can then see these methods used, as well as FathomNet being valuable. And I want to say that we've already integrated these algorithms. So it's one thing to use these algorithms on data that's already been collected. It's another thing to use these algorithms in real time. And so this is something that we've been really working on, thanks to the National Science Foundation's support, trying to integrate these algorithms with vehicle controllers so that in cases where we might have an animal or multiple animals intersecting or running into each other, we might be able to robustly follow them and track them. And so all of this work, again, is being conducted in situ using our remotely operated vehicles. And you'll notice here, in this case, we're tracking Bathycordius, so that larvation. And you'll notice as the larvation runs into or intersects with another animal, pay attention to the bounding boxes as well as the classifications or the labels. And you'll notice that since our target animal is Bathycordius, in this case, we're able to continue tracking it even when the animal is either occluded or overlapped by another. And what we've been able to show is that by integrating these kinds of algorithms with the vehicle controllers, we can have more robust tracking. And so one example is the fact that we've had our longest duration observation of more than 5 hours of a siphonophore. And if you look at just the blue curve over this period of time of 5 hours, you'll notice that we've tracked that siphonophore from 300 meters deep down to 500 meters deep. And you can also see variability in that signature, where not only does the animal sink or swim downwards, it also does this in intervals over time. And so this is part of a project that we're working on with Jack Costello and Sean Colin, again supported by the National Science Foundation. But I want to say that this is really only the beginning, and this is something I'm really excited about. The fact is we can use these models, again, facilitated by these open source resources. We can use them in real time and then pre-program our vehicles to search for and find life or features we want to study or sample. And so this video is my final video. This is from a demonstration earlier this year. And in the top left, in 2021, in the top left, you can see the bright colored text showing the mode that our vehicle is currently in. And as we come up on a giant larvation, the animal that we're searching for, the vehicle automatically transitions from search, acquire to track.
Batho coming up.
Are we going to get it? And we're watching this live as the vehicle comes up on the animal. we transitioned to acquire, and now we're tracking. And so algorithms like these can really revolutionize how we deploy vehicles to explore the deep sea or the ocean, discover life and valuable resources, but also minimize our impact during sampling. And all of this is enabled by open source code, open source resources, and open source data sets that we're trying to build thanks to FathomNet. So if any of this is interesting to you or might be of interest to someone else or other students, there's many of different opportunities available where people can engage with our lab, the Bioinspiration Lab at Mbari. One of them is the fact that Mbari has a summer undergrad research program or an intern program. There's also NSF's intern program, which provides opportunities for graduate students to spend time in non-academic settings, I think up to 6 months after they complete their PhD work. And there's also a very competitive Mbari postdoctoral fellow program. And if those aren't interesting or just don't apply to you, we're also interested in building our collaborator network through the Deep Ocean Inspiration Group. So trying to provide opportunities both at sea, but also utilizing the technology that we're creating for organismal biologists, engineers, artists, and technologists to really think outside of the box and try and study these non-model systems in a really difficult to access place. And so with that, as the FathomNet ecosystem grows through contributions from the community, I really hope it could help unleash artificial intelligence to help us explore and discover our ocean. And we're very fortunate that we're really just building on open source tools that are already available in the computer science community. And I look forward to seeing where this takes us. So thanks for your attention, and thank you to our funders for making this work possible.